Welcome to the presentation on amyotrophic lateral sclerosis, or ALS.
First baseman Lou Gehrig hung up an amazing mark by playing in 2,130 consecutive games. Then a fatal disease attacked baseball's Iron Man. In Yankee Stadium, touched to tears by the tribute, Gehrig made his last public appearance.
For the past 2 weeks, you've been reading about a bad brag. Today, I consider myself the luckiest man on the face of the earth. When you look around, wouldn't you consider it privilege to associate yourself with such a fine-looking man as is standing in uniform in this ballpark today?
It's most popularly known as Lou Gehrig's disease because of the death of the Hall of Fame baseball player Lou Gehrig from the New York Yankees in the 1930s. Yet another name for this is motor neuron disease, as it is called in Great Britain. The actual definition of ALS is a progressively degenerative motor neuron disease. Amyotrophic means no muscle stimulation. Lateral refers to the position of the motor track within the spinal cord, and sclerosis refers to the hardening of this track with the degeneration of the axons. Here is a comparison of a normal neuron with its supply of muscle versus a degenerating neuron and its supply of the muscle. As you can see, there is thinness of the axon as well as the thinness of the muscle. When there is no stimulation for muscle contraction, muscle structure becomes smaller, and that is what is shown in here. In order to understand the disease, we need to go back to the neurophysiology a little bit. The first thing is to look at motor unit. A motor unit is actually a motor neuron originating in either the brainstem or the spinal cord. In this case, they have shown the origin of the lower motor neuron as the spinal cord, its axon terminals, and its supply of skeletal muscle fibers. So one motor neuron and its supply of so many muscle fibers is one motor unit. There are several motor units that are shown using various color codes. The other topic we need to understand is the neuromuscular junction. It is the junction between a neuron, a motor neuron, and the muscle fiber. So an action potential traveling through the motor neuron, reaching the axon terminal, which is enlarged, and this enlarged portion of the axon terminal is called the synaptic knob. The enlargement is due to the storage of neurotransmitters in the form of vesicles. So the action potential triggers the release of neurotransmitters from these vesicles, and the neurotransmitter is in the synaptic cleft, the space between the two cells. Some neurotransmitters bind to their specific receptors on the muscle fiber, triggering a new action potential within this cell. which then leads to muscle contraction. Next, we will look at the details of corticospinal pathway. This is the one that is actually involved in ALS. Corticospinal pathway is a motor pathway. Origin of this pathway is in the cerebral cortex. To be more specific, its neurons are located, the cell bodies are located within the primary motor cortex. Anatomically, this region is precentral gyrus. The cell bodies here are pyramidal in shape, and that is how it's also called as the pyramidal system. So the pathway travels down the brain stem, and in the medulla, a majority of these axons cross over. That's what is referred to as decussation, and then it reaches the lateral portion of the white matter. The rest of the pathway, about 10 to 30%, does not decussate here in the medulla, but continue to descend into the spinal cord. And the location of this is in the anterior white column, which eventually decussates. So the first neuron in the pathway is referred to as the upper motor neuron, which then synapses with the lower motor neuron, which is shown in green here. Lower motor neuron is the one that ends in the skeletal muscle. Let's learn some more details about the alpha motor neurons. Alpha motor neurons are one particular type of motor neurons that originate in the spinal cord. This is the lower motor neuron that supplies the skeletal muscle. Yet another type is the gamma motor neuron that controls the sensory structure, muscle spindle. The alpha motor neuron is the one that receives input from the upper motor neurons from the precentral gyrus as well as other areas. Loss of alpha motor neurons or disruption of their axons could cause lower motor neuron weakness. If there are no alpha motor neurons, that is when there is less signal reaching the muscle, lower motor neuron weakness. Here is a cross-sectional view of the spinal cord showing the details of the lateral corticospinal tract location. This is the lateral portion of the white column, and then the smaller bundle is the ventral corticospinal tract. Let's now look at the key features of ALS. This is the most common form of progressive motor neuron disease, and the death is typically due to the respiratory paralysis or complications such as aspiration related to swallowing. One of the key features of ALS is that both upper and lower motor neurons are involved in this disease. There are several diseases where there is just the upper motor neuron or just the lower motor neuron is affected, but ALS is where both are involved. So if there is not a clear involvement of both of these neurons, one should question whether it is really ALS. Now, looking at the corticospinal tract once again, we need to look at the loss of both upper motor neuron, that is shown in red arrows here, starting with the primary motor cortex, descending down through the medulla. Most of the axons crossover in the medulla. A small percentage of the axons continue further down as the anterior white column. So both are involved. Upper motor neuron, whether it is lateral or the anterior portion, both are affected. And then the anterior tract deposits in the spinal cord, and both the lateral and the anterior spinal tracts synapse with a lower motor neuron, which then ends in the skeletal muscle. Even the lower motor neuron is affected by the disease. So now we look at what are the effects of the loss of just the upper motor neuron, and then the lower motor neuron, and then the loss of muscle function. So basically, you're looking at this entire pathway, taking a small part at a time and explore what is lost from this upper motor neuron weakness or lower motor neuron weakness or the muscle weakness. So when there is upper motor neuron weakness, if this is how it starts, you would see that the number of motor units that are activated is not affected, but there is a decrease in the maximum discharge. So the action potential generated is not the maximum discharge frequency. Okay, so looking at the lower motor neuron weakness, you're talking about alpha motor neuron here. There is a spontaneous discharge from the alpha motor neuron. And this is what we call as the reason for twitches or fasciculations that a patient develops. Additionally, the denervated muscle fibers, so the muscle fibers that are not receiving the input from the alpha motor neuron, discharge on their own, and they could also trigger those twitches or fasciculations. And with lower motor neuron weakness, there would be recruitment of more motor units, and there is a need for more of these to generate the same strength as before, and this will be delayed or reduced because of the loss of motor fibers gradually. When there is a muscle weakness because of the lack of stimulation, you would see that the number of muscle fibers activated will decrease, and decreased contraction force related to this, and the number of muscle fibers activated will be reduced. So there is a decrease in the stimulation, decrease in the muscle force, decrease in the number of muscle fibers responding to any stimulus. The size of action potential will also be diminished, and more motor units are recruited to produce the same power, same force of contraction. Now, looking at the details of what kind of changes do we see within the neuron itself. So this is more of a microscopic detail, and we have already noted that there is the shrinkage of the neuron, smaller size of the neuron, and often that goes with these pigmented lipid deposits. Lipofuscin are the deposits that are often seen in these neurons. Secondly, there are these large structures that appear, and they typically have proteins and neurofilaments. These are cytoskeletal structures that are accumulating in a certain way in large numbers. That could be the cause of sclerosis, the hardening of the tract. In addition to all of these changes, there is increased number of these neuroglia, specifically astroglia and microglia, the two types of glial cells we normally see in the CNS. And all of this, of course, leads to the death of these neurons in a progressive manner. Can the muscle be reinnervated? Is one thought. And in the early phase of the disease, yes, there are some shootings distally that appear, and eventually that is reduced. And in this disease, reinnervation and the recovery of these muscles is not the case. So there are very few incidents of stabilization or regression. There are about 5% of ALS patients who have lived beyond the 3 to 5 year of the survival rate. The 5% of the patients have lived actually about 20 years or longer. So the incidence is about 1 to 3 per 100,000, and the prevalence is about 3 to 5 per 100,000. So the number has been stable all along. It is notable that higher prevalence is in the Western Pacific, like New Guinea. Risk factors are difficult to list, but some of the risk factors that are speculated are pesticides, insecticides, or smoking. There is a question often asked whether it is related to physical activity. Some of the patients who have suffered from this disease are fit and typically never had any major disease before, and they were even active in their physical activity. And there is no real evidence to support these notions at this point. So in addition to the loss of neurons, upper and lower motor neurons, and the muscle loss related to the lack of these neurons, there are other effects that we see in ALS. One of them is to do with the glucose metabolism. There is a glucose tolerance test that is conducted where there is the time is about twice the normal person's glucose tolerance test. And another additional thing that is interesting is the presence of ubiquitin in non-motor systems. So a marker for degeneration, ubiquitin. So this is found in other tissues. So that is interesting as well. So another interesting fact about this disease is some functions are not affected. One of them is ocular motility. The eyes function perfectly fine, even in the late stage of the disease. Of course, the involuntary muscles, the parasympathetic supply from the spinal cord, for example, is not affected. The sphincters regulating the bladder or the gut are still functioning normally. And sensory functions and cognitive functions are not affected either. If dementia is connected to this disease, this could be just a coincidence, but not connected to ALS. So the median survival is from 3 to 5 years for this disease, and it affects men more than women. That's another interesting feature. And typically, this affects in men about 40 years plus. There are some examples, however, where this has occurred in men as young as 20 years of age. 5 to 10% of this is related to mutations, a genetic cause for the disease. So here are two famous people, Jason Becker, the guitarist, and Stephen Hawking, astrophysicist. Both were affected by the disease as early as when they were in early 20s, and both have lived beyond 20 years of duration with ALS, and both are amazingly gifted people. So what is it that it tells us? We do not know at this point. One thing for sure, especially with Stephen Hawking, is this constant care that he receives. He's still working today, and he has been given constant care. And would that be the kind of thing that is needed for these patients, is it to be understood. So the symptoms start off with asymmetric weakness. Typically, the weakness in the hand, weakness in the leg, distal limbs are the ones that show these symptoms early on. Then the unwanted movements, typically in the mornings, that is like jerky movements, or muscle cramp is another thing. Then the muscles become smaller, and that is another sign that there is progressive wasting and atrophy of the muscles. Spontaneous twitching of these motor units, and it is interesting to note that the extensor muscles are affected in the hands, that is the forearm, then the flexor muscles. Then if this disease affects the upper body parts first, if the brainstem portion of the neuron, that is the upper motor neuron, and the origin of the neurons from the brainstem affecting the face, the head, and other upper body parts, then it affects swallowing, chewing, functions of that sort, movements of the face and tongue. If it is the spinal cord, that is the lower motor neuron loss, then it would affect the limbs first. So depending upon where the origin of the disease is in the beginning. So we talked about the hyperactivity of the muscle stretch reflex, those jerky movements, and resistant to passive movements, that is like the muscles that are involved in maintaining muscle posture. Patients with reflex hyperactivity often complain about muscle stiffness more than the muscle weakness. So as it progresses, once it affects the respiratory muscle, especially the diaphragm, death could be due to the paralysis of these muscles. There is the organization that is a committee of the World Federation of Neurology. It has some guidelines for ALS that definitely suggest that it has to be both upper and lower motor neuron involvement, and we have to exclude all of the alternate diagnosis. And if 4 of the parts are affected simultaneously, it is a definite ALS. Probable is when only 2 sites are affected at this point, and possible is when just 1 part is affected. The genetic form of ALS is typically the autosomal dominant trait. Then the symptoms here are no different than the one in sporadic ALS. Mutations of various genes are possible. There are a key number of genes that I have mentioned here. The main one is the superoxide dismutase SOD1. About 20% of the genetic form of ALS is from mutation in this particular gene. Then a small percentage from TDP43, that stands for TAR DNA binding protein gene. Then the other mutation is in this protein, first /TLS, that is fused in sarcoma or translocated in liposarcoma. So we, at this point, do not know what causes ALS. There are some mechanisms that are drawn. One of them is the buildup of glutamate, excitotoxicity. Typically, as glutamate is released, glutamate is an excitatory neurotransmitter, correct? So the release of neurotransmitter at the synapse is usually cleared by the nearby astrocytes using the transporter EAT2. However, in patients with ALS, the number would be reduced for these transporters. So there is a buildup of glutamate, and that could lead to toxicity of this particular neurotransmitter, this particular excitatory neurotransmitter. And in the genetic form, mutation of SOD1, this particular enzyme is involved in the removal, detoxification of free radicals, which simply help astrocytes survive and keep up with their normal function of removing glutamate from the surrounding interstitial fluid. However, mutation in this particular gene would impair this transport, and that is when the astrocytes would have a large buildup of these free radicals, and that leads to the death of the cells, and then, of course, that leads to the toxicity in the interstitial fluid, leading to the death of the neurons nearby. What kind of treatment do we have at present? Very limited. There is no treatment to stop the process of ALS, the progressive nature of ALS. There are a few drugs that have been tested with very little benefit at this point. One of them is riluzole. This is the one that reduces the excitotoxicity by reducing the release of glutamate. If you block the release of glutamate, there is less to deal with, and that would be beneficial. But it only has shown a modest lengthening of survival, about 3 years more than the normal duration that is expected with ALS. The second one is ceftriaxone. This drug is supposed to enhance the transportation of glutamate by the astrocytes. So that is another approach that has been used in these two drugs. Most of the focus is on helping these patients move on with normal life as much as possible. So there are lots of these plans, both for the foot, the finger, then the respiratory support, gastrostomy, that is the external tubing that is placed to supply normal amount of fluids and nutrition, and speech synthesizers to continue with their ability to speak. Well, that's the end of this topic. These are the two references I used mostly to prepare this presentation. If you have any questions about this presentation, please let me know. I'll be happy to answer your questions in class. Thank you.